Welcome, everyone. I'm very happy to see you guys and also people online back for our colloquium here, jointly hosted by the Schmidt Center here at the Broad Institute and then Department of Electrical Engineering and Computer Science at MIT. And, as you know, in the series, we always want to highlight people who either come from machine learning, build foundations of machine learning and bring that into biology, or come from biology and develop new machine learning methods and really rethink machine learning methods in order to make big progress on very important problems. And so I'm very excited to have Grégoire Altan-Bonnet here with us today. So Grégoire did his PhD at Rockefeller University, and then he was for about 10 years at Memorial Sloan Kettering, and after that joined the Laboratory of Integrative Cancer Immunology at the National Cancer Institute in 2016, so has already been there for nearly 10 years again, where he serves as Senior Investigator and Deputy Laboratory Chief. He has a really cool name for his group. It's the Immunodynamics group within the Laboratory of Integrative Cancer Immunology at the NCI, which is really tackling one of the most fundamental questions in this space, namely how the immune system reliably can distinguish between self and non-self and still be super flexible at fighting tumors, infections, etc. For this talk here, he really thinks very heavily about building experimentally validated quantitative models to make great progress on enabling precise, personalized immunotherapies. And in particular, I'm sure we'll hear a lot of really nice examples of this, of how to combine mechanistic modeling with cutting-edge machine learning to really enable these types of progress, connect signal transduction all the way to gene regulation, cell differentiation, cell fate decisions across spatial and temporal states. So I think we're in for a treat. So Grégoire, it's all yours.
Thank you, Caroline. So it's my great pleasure to be here today. I'm going to stand here not to get the light. I'm going to tell you a little bit indeed on what we do in terms of trying to build models of immune response, specifically in the context of cancer immunotherapies. And the joke I always make is that if it starts to sound like French cooking, you stop me and you ask questions, right? So I have a bit of an accent, obviously. Oh, it's not working. OK. So we're interested in really modeling the way the immune system will start to interact with the tumors and really be able to understand how this combination of different cell types, from cytotoxic T cells to myeloid cells like macrophages and so on, they all communicate with each other, they all interact with the tumors to decide whether they go and fight or whether they are going to really tolerate it and let the tumor grow. In that context, there are many different approaches that people are using in the field. And one thing that I want to point out is that there's a classical, more machine learning-driven top-to-bottom approach where people are going to take samples from tumors, from patients, from the blood, from different organs, and try to build a kind of a map and an atlas of all the possible states of activation of these cells, and at some level define some biomarkers and try to capture the network of interactions of these cells to really be able to predict where it's going to go in terms of differentiation. And then there is the perpendicular approach is more bottom-up, kind of more physics-based, where you start from very mechanistic model. You really try to understand how each cell type interacts with each other based on very detailed nitty-gritty interactions map of the cells. And then you can build some experiments and then do biochemical modeling in such a way that at the end you reach a level where you can have some very clear predictions where you can really explain how you can manipulate these cells to really be able to drive the response to the tumors. So of course, both approaches have their plus and minus, right? One captures the whole complexity of the system. The bottom one is more mechanistic and maybe makes predictions. And we want to live at the intersection of the two, where with some math, we're going to be able to capture the complexity of the system, and at the same time try to model these very low-dimensional predictions from the bottom up to make predictions. And our shtick is to really use a bit of modeling to do that. So if you come to the NIH and if you come to many clinical centers across the world, there's a lot happening in immunotherapy these days. This is a classical protocol happening in the surgery branch at the NIH, where people go in the patients and harvest by surgery the tumors or the metastasis, dissociate these tissues, and then grow the T cells, and then with some conditioning where you kind of remove most of the immune system of the patient, you reinfuse these cells, And you hope that you have really selected enough of these leukocytes, you have picked up enough of these T cells and so on, that when you grow them to a huge number of cells, which is typically like 1 billion to 10 billion cells, you're going to be able to put the tumor in a checkmate and do some regression. And this example on the right, you are looking at a primary visceral metastasis, so something very hard to treat. This is back in 2022. This patient came in with stage 4 with a massive tumor mass. And you see that after the injection of a T cell, they were able to regress these tumors. And the patient has been in remission since 2020, I believe, and still doing well. So again, the promise of immunotherapy is that, again, if we are able to manipulate T cells, if we understand fairly enough how these T cells can target tumors, we could have a very good therapy, which would be very personalized for each kind of tumors. But at the same time, when you go to the Rosenberg group and others at NIH and other clinical center, you will see that when these things work, they work extremely well. So the patient really goes in remission, and very often we never see them again. But unfortunately, it works for maybe 10, 20% of the patients, and we don't have a general model. So we are kind of living in that space where we have access to some of these samples, and we are trying to characterize them again in terms of defining how diverse this population of leukocyte can be and how it maps into different response. But for today, I'm going to show you a little bit more abstract thing to be able to develop models of T-cell activation. And I'm going to do it in 2 parts. In the first part, I'm going to show you how we use robotics to really probe T-cell activation, build models of T-cell response, and design better T-cell therapy. In the second part, I'm going to show you how these reactions of T-cell recognizing the tumors can be very stochastic and how we can learn about that in this context. So I start from a very basic aspect, which is like if you go to an immunology lab, it's fairly easy for us to really collect T cells, tumors, dendritic cells, or macrophages as antigen-presenting cells. And when you mix these 2 cell types together, the T cells are going to recognize something on the surface of the antigen-presenting cell, which is called an antigen. And they're going to start secreting cytokine, they're going to start getting activated. So very old data from the 90s shows you that when you titrate the amount of antigen on the surface of these cells, and you measure different readout like IL-2 cytokines or interferon gamma cytokines, this looks like for the same antigen, you're going to get very different input-output relationship, right? So that means that, again, when you look at the signaling cascades triggered by the T cells, it's going to read the different amount of antigens with different set point and do different readout. And this we understand fairly well, which is that we had a mechanistic model in collaboration with Paul François back in Montreal, where we are looking at the TCR interacting with antigens, forming a complex and getting phosphorylated once, twice, multiple times. And all these different stages of activation of the TCR maps into different response. For example, if you want to secrete interferon gamma, you need very little phosphorylation. If you want to secrete IL-3, you need to really go to all the end chain and really activate fully the TCR. So this is kind of the premise of what we have been doing in the lab, which is to say that when we look at the T cell, it's obviously a very complex cell. In terms of signalosome, when you look at the gory details of the signaling cascade, there's a lot of components that we know, a lot of kinase, receptors, phosphatase involved. But at some level, we can abstract it out and make it like a very stage level of activation. And because we know that it really corresponds to different input-output relationship. So at some level, we see something to really resonate, I hope, with this crowd, is that we can really think about the way T cells in red here are able to use their TCR and recognize different antigen on the surface of tumors or antigen-presenting cells as really kind of a signaling bottleneck. It's really an antigen encoding where, again, one receptors pick up different signatures on the surface of the tumors, project it into different signaling pathway, which activates many different cytokines, which get secreted and maybe feedbacks. But at some level, there is really this idea of signaling bottleneck, where the cell is going to pick up the signaling cascade from receptors and project it into a very complex case. So we wanted to take this inspiration really based on machine learning to say that we wanted to be able to really capture a lot of data. So for example, these output in terms of cytokines, but you could look at surface markers, you could look at transcription factors. Whatever you can measure inside the T cells is kind of a readout of the level of activation. And we wanted to collect a big data sets to be able to really start to build a model from scratch. So to do that, we built a platform which is called Immunotron. So again, if you go to an immunology lab, it's fairly easy to get some T cells from, let's say, some mouse model, to get some macrophages, dendritic cells from another mouse model, and then you can mix these things inside a small dish. And we built this kind of robotic platform to be able to do everything you would do at the bench, be able to assemble these reactions, let them incubate, collect supernatant, and so on and so forth, in such a way that we can really do this thing at scale at very different time points. And then when we collect the sample, we can really characterize with a high-dimensional space of features. So we can measure the cytokine in the supernatant. We can measure at the single cell level by flow cytometry. And again, because it's a robot, you can really acquire a lot of time points and get a lot of cells very quickly. So let's just show you how it behaves in the lab. So this is the robot in action. So typically, you have an incubator on the right. We have assembled these reactions like already. At every time point, the robot goes and picks up the plate. And it's going to do everything that we used to do by hand like 10 years ago. And it's going to really go and collect, remove the cover of the plate, pick up some tips, go and collect some supernatant, process the cell for flow cytometry, and then store them for acquisition later. So this is fairly easy to build. If you're interested in this technology, reach out to me. It's not very expensive, but really it enables you to really acquire tons of data very quickly. On the right, I'm showing you a simple Python pipeline which we use to capture the data. Meaning, like in a typical experiment, we're going to have different TCR, different T-cell number, different antigen, different concentrations. And with a simple graphic interface, we can really annotate this is 24 96 well plates. And you can annotate each well, such as you end up with a huge data frame where every single cell is going to be annotated for the type of conditions we have and the type of features which should be collected. And what we get is we get very quickly this kind of big spread of data. So this is almost 5 years ago already, where here on the x-axis, I'm going to show you different features that we measure, for example, different stage of activation of the T cells or different cytokines. And in this training set, what we have is different quality of antigens, which are like kind of a peptide which stimulate the T cells, different quantity of antigens, different time point. And you see that if you do like just a cluster map, there is no rhyme or reason. You cannot really organize the data in a very sensible manner. But if you work in an immunology lab, you will know that they are going to care a lot about these clusters because some of them are going to be memory type of T cells, some of them are going to be effector type of T cells. So it's just to tell you that you can really get a lot of diversity in the way this reaction occurs. But again, if you don't organize the data in a smart way, it's not very useful, not very practical. So the analogy I make is that usually it's meant to be a bit offensive to the immunologists. So I don't know how many immunologists are in the room, but I think that classically when we look at immunology papers, we really have a view of the immune system as kind of pre-Copernicus, right? We are representing all the variables that we are measuring in a way which is very quantitative, but we are not organizing the data very well. So this is the solar system before Copernicus, or maybe the solar system after Trump, I don't know. I mean, there's a lot of things happening these days. But obviously, if we have the right set of variables and you put the sun in the middle of the universe, things are way more manageable, easier to model. Now you can fly to Mars if you're Elon Musk, I don't know. But no, you can really have a more manageable model to deal with. Well, it turns out, and actually that's a paper from Zurich, Caroline, there is some VAE type of architecture that people have used to try to do these things automatically. So in this paper, which is a PRL from 2020, we just fed all the angles that people measure on the solar system, fed it into a VAE type of architectures, and then right away they found that the most compact representation for this set of variables was to really place the sun and measuring all the angles from the sun, telling you in a way that you can have a better model if you organize the data in a proper way. So we want to do the same in the context of immunology. Of course, we don't have Newtonian mechanics, so we don't know exactly what variable to look at, but we hope that with some machine learning, we can get there and really build a model which is going to be more interpretable and easier to manage. So this is, again, what we did with this cytokine data. So again, we take T cells, we activate them with different quality of antigens, different sequences of peptide, different quantity of antigens. We have different type of APCs and so on and so forth. Put these cells in the robot, collect the cytokines, a bit of pre-processing, and we feed them into a very, very simple neuronal net, which is a one-layer perceptron. But what we do is that we really try to organize the data according to the sequence of the antigens. So what we are stating in a way, the working hypothesis here is that when we look at this very high dimensional space, so here I'm just showing you 5 cytokines, but we go all the way to 27 cytokines, we expect that we can find patterns which are purely related to the quality of the antigens, meaning that the T cells are able to really recognize different type of antigens independently of quantity, independently of the context, and so on and so forth. So again, you feed it into this perceptron, and we make sure, and you're going to see that over and over again in this presentation, we make sure that the latent space is really low dimensional because we really want to have something we can look at, interpret, and manipulate back in the lab. So this is what the latent space looks like in this particular context. Again, time dynamics collected from the robot, and here I'm color coding according to the different quality of antigens and different style for different quantity of antigens. What you see is that all the very good antigen in blue seems to be there. All the stuff with very weak antigen is on the left side of this panel. And you have all these trajectories which seem to be very easily parameterized and very easy to quantify. So we built a simple model, mechanical model for these trajectories. Turns out a single parameters, which is really the speed at which you get going, seems to parameterize, is all you need to be able to get all these different set of trajectories. And now when we plot these parameters, which we call immune velocity, as a function of the antigen quality as measured by some other assay, like by doing a dose response, we get, again, from the training set, which are the circles, to the test set with different TCRs and different type of mice, something which correlates very nicely. So what it tells us is that, again, when we look at immune reaction, if we measure enough features on the T cells, in this particular bulk cytokines, and project it into a low-dimensional space, we could end up with a very smart parameterization of the way these T cells are activated, which correlates to what we knew beforehand. But again, we don't need to work very hard. We don't need to purify the cells. We don't need to do a lot of work to be able to define this variable. And indeed, that's what we are using now in the clinic to really be able to rank different T cell activation. Not only that, but the training set was done in a mouse model. And when we do the same with clinical samples derived from T cells purified from the blood of individuals, endowed with a different TCR, and then we test them with different quantity, you see that the model that we derive in mouse, we didn't have to retrain anything. We just look at exactly the same parameters in terms of projecting the cytokine into this very low dimensional space. And again, this immune velocity becomes a very good measure of antigenicity, and we don't have to do anything. So this is very striking because it tells us that in bulk, when we have a pure population of T cells and we look at the way they respond and do different patterns of cytokines, they really reflect different classes of response with different level of antigenicity in a very separatable manner and different class that we can quantify. So this is published 3 years ago. Now we can do the same with single cells. So in the same reactions, now we can collect the cell, send them with a panel, I think 30 markers or so, and we collect, like typically in one experiment, 10 million cells. If you do a UMAP to just represent, again, the state of activation of the T cells as a function of the quality of antigen, you see, again, no rhyme or reason, a lot of overlapping between these different conditions. You can look with different embedding like FATE or PCA. It doesn't make too much sense. But again, when you start to, I'm going to skip that part, if you do a better architecture where you're going to feed the different time point into a two-layer perceptrons, and again, feed it into a two-dimensional or three-dimensional latent space, do it in a supervised manner according to the antigens. If you take a single time point, you start to see that things start to separate very nicely. Because you start to see that a very good antigens is going to be there, a very poor antigen is going to be there, and you start to be good at, sorry, that's the opposite. Very bad antigen is here, very good antigen is here. You start to have a good enough separation which distinguish, again, maybe 3 or 4 states of activation of the T cells. And now if you take the time series and you concatenate these time points, you end up into an even better latent space where suddenly all these different antigens seems to be living in very different place, and you can really classify your response in a very nice manner and define maybe 7 states. And now we are up to, I think, 12 different states with different antigens. So what it tells us, again, is that if you get enough data and you measure enough markers on the side of the immune cells, and if you use very simple machine learning to be able to squeeze the data in a kind of a bottleneck which requires a low-dimensional latent space, you could really end up with a very nice map where things seem to organize very nicely, and you can really separate all these different activation into a very different way. So we live in that latent space now. If you come to the lab, you're going to see that the graphs that most people have on their desktop are this kind of 2D latent space because it really enables us to organize the data. Here you see that a simple 1D graph where you parameterize by the angle, this one being an outlier, seems to be a good way to represent the data, and you can really start to do some math in that space to be able to learn what the T cells are doing. So this was, again, trained with normal T cell, with the mouse models. Now we can move to more complex immunotherapy. And if you are familiar with the field of cancer immunotherapy, you might have heard about CAR T cells. So normal T cell is very complex receptors with a TCR, T cell receptors, a lot of signaling associated chains. And then a normal T cell to really be activated needs this CD28 co-stimulation. Well, at the end of the 90s, a group at Weizmann Institute created these chimeric receptors where they use an antibody on the extracellular domain and then a chimera of the CD28 signaling domain and the TCR signaling domain. And then you end up with something which is endowed into the T cells where it can recognize some ligands on the surface of the tumors based on antibody, and it can activate the T cells in a way which is semi-realistic. So again, this is a bit of a chimera, something a bit artificial. We wanted to see in our model whether it made any sense. So we trained our model with normal T cells, measured the cytokine secretions, then made these CAR T cells, did the same kind of measurements, and see if it projected very nicely. And indeed, even with these very weird receptors, we were able to line up the CAR in black with all the different levels of activation of the T cells. But not only that, we could quantify and make the point that when we compare different levels of activation of the TCR with immune velocity, the CAR T cell lived there, which is kind of one of the weakest antigens we could expect in the case of the normal T cells. So this is very insightful for clinicians because it tells us that, yeah, this CAR kind of work in the clinic, but there's a lot of improvement that you can do in terms of making them look like normal T cells. And indeed, we can work and understand how we move from this kind of CAR simple design to something which looks more like a TCR. But not only that, as we are training this model, we started to do an experiment where we mix both TCR signal and CAR signal. And what we find is that the activation, especially like with these E1 antigens, is lower than what you expect if you had the CAR alone, right? So in a way, this TCR signal associated with this very weak antigen looks very much to like the something intermediate between the CAR alone and the E1 alone. So it tells you that you can really antagonize the response of the CAR in a way that we didn't expect, and again, it looks very nice when you very quantifiable when we're in this latent space. So this gives us the idea to really design a new type of immunotherapy. So again, if we engineer our T cell to have both a TCR and a CAR, we can ask what's going to happen if we suddenly we feed a strong signal in red to the TCR or a weak signal in blue or no signal at all. Are they going to interact with each other? Is there going to be a crosstalk between the two? So in vivo, when we did this experiment, we injected a leukemia inside the mouse. We injected the OT1 CAR with again a TCR and a CAR, and this leukemia express different antigen for the TCR. So if you have tumors with just the CAR antigens, you get this kind of level of death, and the mice die very quickly within 30 days. If you place this red antigen, which is very strong, you're going to have both a strong signal on the T1 and a strong signal on the CAR. You can protect all the mice, and they're absolutely cured. But when you have something intermediate, you would have expected that maybe you get something intermediate in terms of protecting the mice from the cancer treatment. But in fact, experimentally, we get something completely paradoxical, which is that the mix of a weak TCR signal with the CAR does worse than the green curve, which is the CAR antigen itself, right? So this is a big cautionary tale, and again, at the level of machine learning, not too surprising, which is that when you have all this nonlinear interaction between the features, and you project them into a latent space, which is kind of predicting the level of survival of the mice, you can have a very nonlinear effect. In particular, you get this antagonism, which tells us that practically when you have a weak signal going for the TCR, it can really antagonize a CAR signal. Now, this was a complete surprise for the clinicians because nobody had really quantified these things very carefully. But again, with our machine learning pipeline and with our latent space, this thing just jumped out of the data. So the joke I always make is that T cells are very bad at math, right? So if we look at a CAR in green by themselves, they do this level of protection of the mice. If you have a strong signal, 1 + 1, you get 2, and you get a very good signal. When you have a weak TCR, which is maybe 0.2, you would expect 1.2. But in fact, when you mix 1 plus 0.2, you end up with something which is lower than 1, and you really get antagonism. So this is really something we have to pay attention to. It has been completely overlooked in the clinical settings, but this is something we can, again, a cautionary tale for clinicians, and also something useful that we can use to design better CAR immunotherapy. So we went and did that systematically. We call this system EEBS for antagonism enforced breaking system CAR T cells. So one of the postdocs who was on the project is Japanese and he loves cars, so he really insisted in coding EEBS, like a braking system in cars. That was the idea, right? And the idea is that we are going to make CAR T cells with the usual CAR ligands, but we're going to pick a TCR in such a way that the TCR on the T cell is going to recognize the tumor and the neoantigen in the tumors in a positive manner. So which means it's going to really accelerate and synergize with the CAR to really get good killing of the tumors. But the same TCR is going to be chosen in such a way that it can recognize the self-antigen on normal tissues in such a way that this is going to be an antagonistic signal, and in that way you're going to be able to protect the tissues. So again, this is very practical and very abstract, but we can really go back and do that in a mouse model. So we have a mouse model where we're going to adaptively transfer some normal cells which are luminescent that we can measure in the lung. We're going to inject tumors on the side to be able to measure on the flank whether a tumor grows or not. And then injecting these different type of T cells, so no T cell at all, only TCR signal, only CAR signal, or IABS, which has both a CAR and a TCR, we're going to be able to compare whether we're able to kill the tumors and whether we're going to go and really kill the healthy tissues. Again, the ideal construct should be able to kill the tumors and let all the healthy tissue very, very much alive. And these are the data. So in black at the top, when we don't do any treatment, the tumor keeps growing and becomes very big. All the healthy cells in the lung are still there, but again, the mouse dies because you're not able to control the tumors. If you are using a regular CAR, which is really the CARs which have been tested in the clinical trial, like a HER2 CAR in pink, you get a very good control of the tumors, meaning that the tumor shrinks dramatically, but all the healthy cells inside the lung gets killed at the same time. So there is not enough specificity in this CAR to get you going. And again, this has been done in clinical trial, and unfortunately, the patients die because you are really losing your lung in this type of CAR treatment. And then our construct in orange, so it's not perfect yet, but we are trying to optimize it. But you see that you have the best of both worlds. You are killing the tumors and controlling the tumors in a very nice manner, and the mouse can survive very nicely. And you don't kill all the healthy cells inside the lung, so in a way the lung gets preserved and you don't get much off-target toxicity in this kind of construct. So this is just an example to tell you that we can really use fairly rich data sets in the way we activate T cells to learn a good model, learn about this antagonism and the crosstalk between different receptors inside the T cell, and use that all the way to clinical trial. So this was published last year, and we are indeed in the process of making a clinical trial to really use this idea. So a lot of people were involved, Suraj in my lab, François Bourassa and Thomas Rademacher in Paul-François' lab for the machine learning part, Taisuke Kondo in Naomi Taylor's lab to do a lot of the experimental work with Suraj. And then when we move to the single cell, we continue with Mahmoud and Madison, but I didn't present many data on that. The key message that, again, you can use machine learning to compress these very high dimensional data sets into something manageable, which really models the way T cells get activated. You learn about the way weak TCR signal can antagonize CAR activation. Then you can design a system which we call ABS again to be able to really harness both sides of a CAR T cells. You can really force them to kill tumors more efficiently. You can help them block toxicity against cell tissues. And again, the key idea is that you can really start to go inside the freezer of NIH and test different TCRs for their ability to really discriminate between tumor and self and build a better model on that. So again, the first part was published in 2022. This ABS system was published last year, and the single cell machine learning is almost going to be submitted very soon, so reach out to me if you want to learn more about that. So that's the first part of my talk. Maybe I can open for questions. Yeah, go ahead. Thank you.
Can you show about how immune? Sure, I'll take the microphone.
Yeah, you don't want me to repeat your question in French.
Given what you showed about how immune activation velocity versus antigen affinity has a one-dimensional monotonic relationship, how do you think this compares or contrasts with findings about how high TCR affinities can drive exhaustion and lack of tumor control?
Yeah, so what we are telling you is mostly high TCR. So in fact, when you look at the classification, you can go back to the features and try to understand what drives this hierarchy. And indeed, when you reach these very good antigens, which are very at the end of our hierarchy, and we know that on a short time scale they give you a lot of readouts. Indeed, they get placed in that very tip of the latent space because they are activating a lot of exhaustion and a lot of blockage. So at some level, what you want the 2D latent space, because if you look at the details in the single cells, you're going to see that there is a very nice linear hierarchy, and then it starts to go back in terms of activation. So some markers get lost, some other markers move you a little bit away from Wendy, and indeed you rediscover the idea of exhaustion by projecting to a slightly higher dimensional space.
Very much.
Yep. Other questions?
Can I ask something about that? Like, okay, so when you're weak, you're antagonistic, but if you go too close to zero, you're right.
Yes.
So what is the actual math?
Yes.
If
So
you
the
become
math
strong,
is,
you're again +2.
So historically, we had really an explicit biochemical model where we really understand these things very deeply. What's happening is, again, when you engage a TCR, it starts getting phosphorylated, moving towards activation. And as it's doing that, it's activating a phosphatase, which is trying to blocking you from moving forward. So the balance between the negative feedback with phosphatase and the kinase, which are trying to activate you, define T-cell activation. It's kind of a brake and an accelerator, right? So if you have an antigen which is like the self, you engage the TCR, but you don't even activate the negative feedback. If you have an antigen which is in big quantity but not very good at binding, you're going to activate tons of negative feedback, which completely doesn't give you a chance to reach the end of the chain in the kinase. So again, this model tells you that self is self. It doesn't activate much. Weak antigens, especially in big quantity, activate a lot of negative feedback and block activation. But vice versa, if you have a small quantity of good ligands, they do activate a bit of negative feedback, but they manage to reach the end of the chains fairly efficiently, and there's not enough negative feedback to block you. So the system is built like that, not that, because you really want to block activation from the self. If you have something which is really weakly binding, and it's the self, it's most likely in big quantity, and that's what you're trying to protect against. The price you pay is that if you have a big quantity of good ligands, going back to the question about exhaustion, you're going to activate so much negative feedback that you go back in a regime where you don't get activation. So that's why every time you look at immune activation, very often you get a nice dose response, and at a very high dose, it starts to tank, right? It's because you get into a regime where all these negative feedback, which are here to protect you from the noise, now that you have a big signal, input signal, they tend to be activated so quickly that they're going to start to tank their response. So that's the price you pay for being able to be good at discriminating. We have mouse model where we can remove this negative feedback. The mouse is still, the T cells are still responding, but you get autoimmune disorders, right? So it's always a give and take between these two pathways. Yep.
Have you tried to interpret the two latent space dimensions, and are these consistent across models? Like, do they reflect different kinds of cytokine programs?
So, I mean, the shocking part, and it's a bit of a dark joke, is that we train on a mouse TCR transgenic RAG knockout model, and then we moved to clinical samples, and we didn't change the parameters. Swear to God, it's exactly the same numbers. So interferon gamma in mouse is interferon gamma in humans. No, IL-2 in mouse is IL-2 in humans. And the latent space worked perfectly well. We are hoping to increase the space of features, the space of conditions in such a way that it's going to break. But so far, we have been very consistent, and we can really project things from one to the other. When we move to single cell, things are a bit more noisy in terms of latent space. We have to do a little bit of retraining and to adjust this latent space. But again, when you start from like a 100-dimensional space and project it in 2D, it gives you a lot of robustness. You can forget some of the markers. You can miss some measurements in time point. And the latent space is fairly robust. So we think we're onto something there.
Do you have a sense what they represent biologically?
Yeah, so again, because it's such a low dimensional space, now you can project every individual marker against this variable. And basically, there are like a bunch of markers with different set point in terms of input-output response. Most of them are monotonous. Some of them are not monotonous. And I can show you some graph, but we can go back and really explain what it is. So for example, for antagonism, we see that again, you have this pathway which is negative feedback and this pathway which is normal T-cell activation. When you activate this negative feedback very drastically, you already kill the CTL response, the killing response of the T cells. You are still going to get the same inflammation, but the ratio of inflammation to killing gets biased toward less killing when you have antagonism. So we can go back and really interpret these things.
Thank you.
This is very interesting. So have you ever gone back to like published cancer vaccine data where you're giving a patient like 10 antigens and some respond and some don't? And do you think...
So we're making our own data sets, but it's exactly why we build this pipeline. In a regime where we have a polyclonal population of cells, and we mix a bunch of peptide. When you project things onto this latent space, you can really rank the quality of your vaccine very nicely. And we are hoping that we are going to be much better at predicting what's going to happen in different contexts. That's why it's a robot. We hope that it's going to be live. And as the clinicians are identifying antigens, we can test live how the patient is going to respond before we even inject. The cautionary tale is that a lot of cancer vaccines, I was mentioning that at lunch, lots of cancer vaccine uses 20 predictions. But you see that there is antagonistic modes, right? So you have to be very careful that it's not a linear system, and we really want to avoid this antagonism, and we think we can do way better in terms of cancer vaccine based on that. Okay, I'm going to keep moving for the second part. And again, we can always discuss more at the end. So in the process of building this robot, we started to really multiplex and do a lot of repeats of the experiments, and we saw an opportunity to be able to really study the noise or the variability you're going to get when you set up this assay in ex vivo and in vitro. So it started again from in vivo data, where we had a very hard time controlling for the noise from experiment to experiment. And Emmanuel Salazar-Cavazos, a postdoc in the lab, set up a very simple assay, where again, we purify some T cell from a mouse model and mix them with a tumor cell lines, in this case, a B16 melanoma cell line, which classical model of immunotherapy in the lab, which expresses the antigens. And then you put them in a robot, and we can really multiplex these things to measure a lot of replicates and a lot of conditions. And what Emmanuel found is that when he counts the number of T cells which are alive after 4 days of this assay, as you titrate the number of T cells, you get more and more killing, so the number of tumors which are alive goes down. And then he noticed that it became very noisy from condition to condition. So this is for naive T cells. This is from effector T cell, which requires much lower numbers to be able to kill. So at 3,200, you already have what you would get at 16,000. But again, the idea is that even though this is really a simple reactions, look like 2,000 tumor cells and a fixed number of T cells, and we are very precise in the way these things are being set up, we end up in a regime where you have almost like a tenfold difference in terms of the killing from well to well. So we represent that with this simple honeycomb. Each square here, each hexagon here represents one well in our reactions. So again, this is just 19 replicates, but typically we do 100 replicates. And as you titrate the number of T cells, you go from a regime where no tumors are getting killed, where practically all the tumors are getting killed and very homogeneously. But most interestingly, when you're in the middle, when you are like 50% of your IC50, you see that some wells let the tumor survive, some wells kill all the tumors, and you have a lot of noise. So of course, as a PI, I would complain to Emmanuel that it's being sloppy, right? And it's not the case. No, Emmanuel is super careful, actually. And he went and did this experiment to prove his point, which is that when you try to kill these tumors with chemotherapy, like cisplatin, doxorubicin, or with the cytokines we think are important to kill, like TNF, interferon gamma, you can get the same level of average killing, but absolutely no noise. It's very reproducible, right? So when you are killing tumors with T cells, macroscopically, you end up with something extremely variable. While you're killing tumors with chemotherapy, it's much more reliable, and you get much less noise. So we quantify that. I'm going to skip that part for the sake of time. And what we quantify that looking at the number of T cells which are activated in well to well, what we kind of suspected that it was really at the immune level that you had a lot of variability. So for example, here we're measuring at 48 hours the CD69, which is a marker of activation of T cells. And each of this line is a histogram for the T cell inside that well. And you see the distribution, like some replicate number 4, none of the T cells are activated. Some wells, like practically like 50% of the T cell are activated, and everything in between. So all the noise that we get in this very simple immune reaction where you mix tumors with T cells is represented at the level of T cell activation. And again, you have all this noise happening depending on the number of T cells. So the graph that we end up with measures the noise as a coefficient of variation, so standard deviation divided by mean, as a function of the mean number of T cell activated. And these are like very preliminary graph that we obtain. What you see is that the dependency in log-log scale looks like a shifted Poissonian curve. What that means is that the CV looks like 1 over square root of n with a shift, meaning that, again, Poissonian means that it's a low number issue. Very few T cells must have been activated, and then they are going to propagate and really activate a lot of T cells. But again, the fact that you have this one over square root dependency means that at the core of this reaction, there's a small number of cells. If you are lucky enough, you have them, you're going to kill it efficiently. If you're not lucky, you don't have them, and then you get a little killing. So the idea is that, again, when we go back to the clinic and we are looking at this bag of T cells which are being transfused inside the patient, maybe some of them contain very highly functional T cells which are going to be very good at killing the tumors and really good at initiating an immune response. Or maybe some bags don't contain these cells and they are very low functionality. It gives you a lot of variability in this response. So again, it's a bit of a number game where when we look at this bag, there might be, again, some of these highly functional cells that we care about, and we're going to call them spark T cells because they are very good at sparking immune reactions. And then some other patients, some other bags of cells don't contain these sparks, and unfortunately, you don't get a good immune response. So the model is very simple. So in terms of Poissonian, right? If you look at the mean number of cells inside this bag, it's going to be alpha, a certain fraction of the total number of cells. The variance is going to be alpha n by definition of a Poissonian, and you end up with a CV, which is one of the alpha is different definition compared to the previous one, but again, a dependency of 1 over square root of n. Again, that means that there is a discrete population of T cell which seems to do a lot of work in being able to initiate immunotherapy. So the model for this spark is like that. In our bag of cells, we have some cells which we call protospark or non-spark. As they are exposed to the tumors, these get activated. We did a lot of work to understand the feedback, which is that they secrete interferon gamma, they upregulate the HLA or MHC on the tumors, and once you do that, then this non-spark get activated, and they get into a regime where they are activated and they start killing the tumors. So at some level, again, you have a low number of these protospark. If you have them, you can initiate the response, get gamma, and get everybody involved. If you are missing these protosparks, no gamma early, these guys never have a chance, and you don't get any kind of activation. So we can really relate microscopic cellular noise inside our bag of cells into something which is really a macroscopic response for the T cells when they fight the tumors. We spent some time validating it. For example, we can upregulate by pre-exposing to interferon gamma. We can block interferon gamma. So again, in a normal setting, we have this sheeted Poissonian where the 1 over square root dependency appears. If you start to block interferon, if you pre-expose the tumors to interferon gamma, you get much less noise because in a way you have done all the work of starting to upregulate the MHC, and then things become way more reliable. And vice versa, if you block interferon gamma, you get much less killing, and again, much less noise. And we can explain all these things with a model. The line that you see here is really a simple dynamic model which can really fit all these parameters based on the biochemistry. So not only that, so again, this is something which is very useful for clinicians because when you go into the clinic, you see that every patient seems to respond to immunotherapy with a lot of variability. Most clinicians will tell you, well, these are different HLAs, these are different type of tumors, and so on. What we are saying is that there is maybe a space with a lot of variability comes from the presence or the non-presence of these protospark and spark T cells, and being able to really go back to immunotherapy and measure where these cells are could really help in designing maybe better immunotherapy. So we do all these measurements in a robot. We have a lot of wells with many different conditions. We test different number of T cells. We measure how much killing we get. Again, big spread in terms of noise. And you end up with a very simple number, which tells us that out of like maybe 2,500 cells that we inject in this sample, on average, 1 cell will be a spark, right? Simply by looking at the Poissonian signature, we can tell you what fraction of the T cell we inject inside these reactions seems to matter and explain the Poissonian dependency that we have here. So 1 out of 2,500. Not only that, now we can go back to our robot and design a single cell panel to look at the T cells. So you can do your usual clustering, your usual analysis. You find that at 12 hours, the number of sparks, the number of cells which are producing interferon gamma match what you expect in terms of Poissonian, right? So you can do it for different quality of antigen, different T cell numbers. Again, we know that this is really about cells producing interferon gamma that we can identify on this UMAP. And then we can use fate to walk back in time and try to really explain where these cells come from. So again, these sparks have a few cells which are producing interferon gamma at 3 hours. And phenotypically, they are very close to these cells, which when we go back to the naive T cells at T equals 0, seems to have a very distinct phenotype, right? So this is our pipeline that we call STOICS. where again, by multiplexing all these reactions across time, across number of tumors, across number of T cells, and then measuring how much killing you get on a long-term scale, you can predict how many spark T cells you have in these reactions. And once you go back and then take these things and measure at different time points their phenotype, you can start to match the frequency of these clusters with what you predict in terms of sparks, identify where they come from, and then be able to really sort these cells and make a prediction. So this is a phenotype which is simple enough where we can sort the cells. And indeed, when we look at this phenotype for mouse T cells, CD5 low, CD11A high are protospark T cells. When you put them in a dish and you add PMA anomycin, within 1 hour, you're going to see that a lot of them are going to make interferon gamma. And when you take the other cell, which is, depending on the mouse, like from 95 to 99% of the cells, when you give them PMA anomycin, you would have almost no activation at all. It would take really 24 hours to get any kind of activation. So this is really some progress because, again, based on our pipeline, we can really go look at the viability of response of T cell against tumors and then walk back. So this is done a bit in a pedestrian manner. We're working with some colleagues to use optimal transport to really be able to identify these things more reliably in a more robust manner. But again, importantly, from the immunology side, we can really identify a subtype of naive T cell, which is there at T equals 0. And these cells are really the one you care about if you want to really spark an immune response. So we did some transcriptomics on these cells. We could not find anything. Only when we moved to attack 6, we found something very interesting. So this was done in collaboration with Josh Waterfall at Curie, where when we look at our protosparks, again, at T equals 0, we sort the cells for non-spark in black or spark in red. And when we look at single cell attack 6, we see that inside, and there is almost no difference between all these cells, except when you look at the interferon gamma locus, and you have a subfraction of these CD5 low, CD11A high cells, which are really good at opening the interferon gamma locus, and they're going to be very good at making interferon gamma very quickly. So again, our gate was not perfect. You see that there are a lot of cells which are maybe in a different phenotype, but at least it gives us a frequency that within this gate, maybe 30 to 40% of the cell seems to be open for business. The interferon gamma locus is open, and that's all you need to be able to really make interferon gamma very quickly. And indeed, when we test functionally this idea, we sort the cells for CD5 low, CD11A high. We expose them to PMA anomycin for 1 hour, 6 hours, sorry, in this particular case. You see that both the protospark and the non-spark get activated to comparable level. But when you look at the interferon gamma at the level of individual cells, only these protosparks are able to make interferon gamma reliably, while all the non-sparks at 6 hours, up to 12 hours, they still have not got started. They got activated by the PM anomycin. It takes them time to open the interferon gamma, and in a way they are so slow that they don't contribute to immune response. So this was done, again, as a training set with mouse T cells. We moved to a more human preclinical setting where we collected blood from 8 individuals. We made T cells by forcing them to express a TCR, which recognizes a very specific antigen. And again, when we mix these T cells with different tumors, we get into a regime where things get very noisy, which is what we need to say that even within this bag of T cells, which are identical with their TCR, you are going to get some spark T cell, which explains the noise in this reaction. We apply our pipeline to be able to predict how Poissonian it is, and then based on this Poissonian signature, predict what frequency of cells we're looking for. So typically, it's going to be, so sorry, I forget the numbers, but maybe 1 out of 10, 1 out of 20 cells is going to be enough to be able to spark this response. We go back to our pipeline, we use FATE, and we identify a completely different phenotype because these are like human T cell blast. But like when you look at some cells which are like CD45 RO positive and CCR7 low, so kind of effector memory cells, these are really the cells which are really making the spark, which are going to be able to predict all the noise that we can measure in these reactions. So again, the same pipeline, but in more clinical settings, tells us that indeed, even the prep being done in the clinic, there is a lot of variability. Even if we are in the best conditions where everything is done very reliably, it's going to be very noisy. And this can explain why there is so much difference in cancer immunotherapy. So the markers are here. If you want the signatures for this spark inside the clinical setting, they are going to be CD39 high, CD2 high, HLA-DR high, CCR7 low. And now we can take these signatures and go back to TCGA we can validate that indeed these are the cells which make interferon gamma very quickly compared to all the other phenotypes. And when we go back to the TCGA, when you start to sort the TCGA for tumors which are low in MHC versus tumors which are high in MHC, and you look at whether there is a protospark signatures or no protospark signature, you see that among the patients we came in the clinic with a low MHC tumors, very low inflamed. If they have the luck to have these protospark signatures, there's a 40% chance that they're going to do well with checkpoint inhibitors. While in the same cohort, the tumor is very cold and very low in MHC, and you are missing these protospark signatures, now you're going to see that these patients don't do well at all, and unfortunately, they die very quickly. In the same cohort, when you look at the patients which come with very inflamed tumors with very high level of MHC, like in this particular, these type 2 genes, all of them do very well, and this spark signature is completely relevant. So what that means is that suddenly we can really go into the clinic, in a way rescue a bunch of patients which maybe the clinicians wouldn't treat because they are very very cold in terms of inflammation, they have very low level of MHC. But if they contain a protospark which is fairly high, they are going to really benefit from anti-PD-1 therapy, and they are really going to be rescued and potentially survive better. So again, this is an example where, I mean, these data are TCGA, right? I mean, there's so much diversity among patients. You are not really powered to do this analysis if you don't have a very clear hypothesis. But what I posit is that our method of using ex vivo models, really studying the key part of T-cell activation, can give us a narrower set of features that we can look after and really power the statistics in a way that we can really go back to clinical data and really explain some of the variability that we observe. So that's it for the second part. Again, this is work which was mostly done by Emmanuel when he was a postdoc in the lab. He moved on to go to big pharma, and with some help with Dong Yajia for the machine learning. This was published like 3 weeks ago, so you can look at all the gory details in there. And again, the idea is that there is a lot of variability in the way these immunoreactions take place. We can use this stochastic signature, this Poissonian signature, to really get a target, quantitative target, which tells us how many cells we are looking for. And the model is fairly simple, which is that these spark T cells seem to exist in a state which makes them produce interferon gamma very rapidly. And that's all you need, this little pinch of interferon gamma to get things going, because you upregulate MHC presentation, and then suddenly all the bystander T cells can really go in and attack the tumors. And we are following up because we think that these spark T cells are really not only a good biomarker, but also something we can manipulate in clinical settings and hopefully boost cancer immunotherapies. I'm going to finish with a little bit of math and a quiz for you guys, right? So at some level, right, we know that a single T cell can secrete 1,000 molecules of interferon gamma per second. This is the numbers we can measure in the lab. So in your mind, if you put a single T cell inside the tumors or inside a small lymph node, how much time does it take before you accumulate enough interferon gamma that everybody's going to see interferon gamma? You don't have to do the math. I'm going to give you the answer right away. Is that it takes practically 17 hours. So you are just trying to make 10 picomolar at the scale of a lymph node. And if this cell is producing interferon gamma at 1,000 molecules per second, within like 17 hours, you're going to make 10 picomolar. Everybody inside this tissue is going to respond to interferon gamma. So a single cell can really impact macroscopically what's going to happen inside the lymph node. Another idea is that when you look at a single T cell, they can proliferate fairly rapidly, typically every 6 hours. But again, within 4 days, a single T cell can really reach a level, and they make 10%. That's why you get your lymph nodes very hard when you have a flu or a cold. They replicate so quickly that from a single T cell, suddenly 10% of your lymph node is going to be made of these particular clones just in 4 days. So this is the power of immunology in the sense that very rare events, a single T cell being activated in the right setting, being able to proliferate, can get you into something very macroscopic, like making enough cytokine for everybody to see, or making enough T cell that you can really go and fight at a macroscopic level. So that's it for the second part. Again, this was the lab pre-Doge, so it was already pretty small. Now it's only 4 of us, so we have been whacked pretty badly, but we are still standing. We have a robot to help us, so it's fine, but it's a bit painful right now. Lots of collaboration, which is a great thing at VNIH. We can get clinical sample from Steve Rosenberg, from Naomi Taylor's, a lot of collaboration with different groups. I would give a shout out to Paul François' group at Montreal, because we do a lot of theoretical group, theoretical discussion with him, and we do a lot of data processing in collaboration. Thank you very much, and looking forward to your questions.
Thank you very much for a really wonderful talk. Questions?
Have you ever looked at like sex differences in protospark abundances,
Yeah.
or looked compared protosparks in people with like autoimmune disease to healthy people?
So no autoimmune disease. Sex didn't matter, but we didn't do it in a very systematic manner. So I skipped that part. Yes. Here we did 8 donors, 8 healthy donors, and we literally like bled all these people in the same time at the same day, 20 years old to 50 years old, and half and half in terms of sex. Prepared these T cells and activate them in our Spark assay, and they are all over the place, right? So some of them are making tons of Spark in terms of this phenotype, and then they make tons of interferon gamma very quickly. One of us, because obviously it was our samples, didn't make much of his Spark and so on. We want to take the same people and redo it, because we think that it's all over the place and it changes over time in a very unpredictable manner. So at some level, there is something in the way our T cells are made. If I take your blood today and take your blood, let's say, after exercise, after food, maybe after an infection, which seems to regulate how many of these cells are in the blood, and it's all over the place. So I think like usual, you might be right that at some level, or let's say an autoimmune person prone to autoimmune makes more Spark, is more prone to making interferon gamma. We know that males tend to make more interferon gamma. That's why we call man flu, right? That's why people who are, tend to be more supposedly more in pain. I don't know if it's really correct, but at least we make more interferon gamma. But that's not really the case here. It's really about how many cells are making interferon gamma. It's very different.
More questions? Can I ask you, so, I mean, the proportion and the type of these Spark cells was dependent on mouse and human, but what about different types of cancers, et cetera? Will it?
So this is from the T cell side, right? So we are definitely interested in checking like different patients because they come with different history, right? Some of them have been whacked with like chemo, some of them had a bone marrow transplant. So we have not done enough of things in that space. I would state that in the context of mice, we have tested, I think, 7 different mouse models, and it's all over the place, right? So even though you put a TCR transgenic and they are all identical as far as an immunologist would tell you, they are all naive T cells. Depending on the TCR, you have a mouse model where like 40% of the naive cells are Sparks, to a model where it was almost like 0.2%. So we don't understand exactly the ontogeny of these cells. Of course, it's very critical to follow up to be able to make them on demand, and that's what we're doing now.
And maybe for the first part, can I ask, so at some point you would like to have an antigen representation that will directly map you to the right place.
Yeah.
How are you?
So we are hoping that in a way what we are capturing in this low dimensional latent space, in a way, is just biophysics. But what was very hard to measure in the past, you had to purify the TCR and then really do a biophysical measurement. But this is just a direct measurement of the K-off of a peptide MHC to TCR. So in that way, we'd be able to connect to more structural analysis where, not AlphaFold, but something a bit smarter than AlphaFold would really be good at predicting how well a given TCR binds to a given antigens. And in a way, this would be a way better measure to be able to train these structural models so that we can really predict. Because when you go to cancer vaccine field, we are very good at predicting what gets presented, but from the TCR side, it's a disaster, right? So again, we had this discussion at lunch. I'm predicting that within 5 years, this problem is solved. Most likely by you, but we'll see.
With TCR representation,
That's
I
right.
think is more.
You wanted a good TCR representation, but I think like there is a bit of acceleration happening because more data sets are being done. We have better measures, right? I mean, there's kind of a virtuous cycle where we are going to get with the right training set, and we're going to get a better sense of the biophysics. I will point out that biophysics is one thing, at least to rank for a given TCR different antigens. But if you go from one clone to another clone, based on their history, it's going to be very different. So meaning that the threshold between self and non-self may be like at 1 second off rate for this TCR and 10-second off rate for this other TCR. So there won't be a universal parameters. It will be very dependent of the way they interact with self ligands, most likely. But it's not hopeless. We think that, again, with this platform, we can really map out on a big scale of how things are happening.
Many thanks again. Towards the goal of mapping some of these Spark T cells in the wealth of human transcriptional atlases, apologies if I missed this, do you think that there's a more broadly encompassing signature than the ENTPD1 CD2, or is that just all you need, those 2 genes, and we're good?
So in the context of the data set we have done, that's all we needed. I think if we had all the epigenetic, if we could really measure the chromatin is really what we care about, right? So surprisingly, at the transcriptomic level, we didn't find anything, but if we could really measure what fraction of cells are really in that state, which is really open for business, you will really have a good signature. So then understanding why, maybe looking at this locus and other locus, there's a Tibet signature, there's a RANKS1 signature, so hopefully we get more upstream, but yeah.
Hello, great talk. I'm curious what your thoughts are on like if the big opportunity in robotics in immunotherapy right now is like higher throughput experimentation and generating larger and larger data sets, or if it's in the kind of promise of adaptive closed loop experimentation where machine learning algorithms are sort of driving like the next set of immune perturbations or that kind of thing.
Yeah, so the second part, I'm not smart enough to really be able to do that, but I think the first part is very promising. So the shtick in the lab is to say we would rather have 1,000 different conditions and a lower level of space of features rather than few conditions and a very large space of features. There is so much biological diversity from patient to patient because of the genetics, because of the type of tumors and so on. But the model you use when you have a low depth in terms of biological diversity is not very robust. So the shtick with the robot is that, again, we can do like 100 times more experiments than we could do like 5 years ago. I'm not claiming that this is a very large data set, but again, it's a lot of cells, right? So 1 week in the lab, it's 1 billion cells with 100 features, and the 100 features are curated based on what we know. But again, in terms of machine learning, 1 billion cells, you can really capture this diversity of cells and so on. So all the outliers, we can really build models which are very good. So from our side, we are trying to robotize things because we want these things to be next to the clinicians. Every time they take a blood sample, every time they start making cells for their patients, we stick them in the robot, expose them to different tumors, different inflammation, see what's in their guts, and then help in predicting what's going to happen. So we don't claim to capture everything what's happening in vivo, but we say that like this stress test really gives you a picture of what's possible, and then hopefully it's a better biomarker and then tells you what to do. So thanks.
Okay, let's maybe end here. So thank you very much, Grégoire, for a really wonderful talk.
Thank you all.